Good afternoon, everybody. We're going to start, and welcome to the TN2 online lunch series of 2021. It's a little bit of a different format, but I think we have a very nice program for you today. So what we're going to do now is I'll give you a few words of introduction, and then we'll move on to the speakers. So it's a webinar in Zoom, as you're probably used to by now. So webinar means that only the speakers and the hosts, that's me, Menno Schoonheim, only we are visible. So all of you listening, you're not visible, so don't worry about your camera, and your video and audio is muted. What you can do is ask questions in the Q&A. There's the button below, and we'll monitor the questions that you post, and the most interesting questions will be asked to the speakers. And you can also vote for which question that we should choose by upvoting the questions that you think are most interesting. Now you can also adjust the way the screen looks. Here's an example. You can enlarge the video of the speaker, or maybe enlarge the presentation by sliding the little bar in between. All right, so a little bit about TN2. TN2 stands for the Translational Neuroscience Network, and our aim is to translate between fundamental and clinical neuroscience. We have annual conferences with alternating topics between neurology and psychiatry. So this time it's neurology, and next year will be psychiatry again. And usually we meet in person, giving you an opportunity to meet each other, but also to meet industrial partners for collaborations. This year, the theme is imaging of immune activation and neurodegeneration in the brain. And it's an online session, as you know now. We have 5 webinars, and at the end of the session, I'll show you what else is still coming. And below here, you can see the coordinators that all are helping me, and we're helping each other to perform this nice session. So now I'd like to invite Mike Wattjes and Rick Ossenkoppele to turn on their cameras and unmute themselves. So welcome both of you. I'll briefly introduce you. Mike Wattjes is a neuroradiologist and head of imaging of neuroinfection and neuroinflammation at the Hanover Medical School. And Rick Ossenkoppele is an assistant professor here at the Amsterdam UMC, specialized in Alzheimer's disease. So welcome both of you. I hope you're excited to get going.
Absolutely, Menno.
All right, so let's start the first poll because we'd like to know a little bit more about you guys as well. And while we wait for that, yeah, so click on the option that is most applicable to you. What is your current role? And while we wait, unfortunately, we as panelists cannot click on anything. We cannot vote, so we have to wait. But the nice thing usually about TN2 is that it's a mixture of scientists, clinicians, and industry. So I hope that the online version will be there as well. Have both of you been to the TN2 before?
So I've never been there physically, Menno, but I watched the first one with the 2 keynote sessions a couple of weeks ago.
Oh yeah, yeah. Normally the physical edition is quite nice. We were at the arena one time and at the AMC another, very nice venues. But now, unfortunately, that's not possible. So the results are in. Most of you are PhD students and principal investigators and postdocs, and also some industry, bachelor, master students. So I think it's a nice mixture. All right. So what we have now is 2 more polls, one for Mike and one for Rick to introduce the topics. So first we'll go to the poll of Mike. RCNS opportunistic infections, white or gray matter disorders. So Mike will tell a lot more about this in his talk in a second. But Mike, maybe you can introduce a little bit how you ended up being one of the world experts on this field.
I don't know whether I'm a world expert in this field, but
I would say so.
I started working during my time in Amsterdam in the MS field, and So we came across the problem with the natalizumab-associated PML stuff, which was a huge issue. There was no data how to diagnose PML, what the imaging features are. So we started from the beginning. So evaluating the imaging findings, developing diagnostic schemes. And apparently we were the first addressing the issue. So we at some moment we became experts. So and we tried to expand our knowledge also to other immune conditions like transplantations. And now I'm back in Germany. We are focusing a lot on the treatment of PML and the role of imaging. So we're now expanding a little bit the role of imaging, not only based on natalizumab-associated PML, but also extending it to other underlying immune conditions.
Yeah, very interesting. And the audience believes it's mostly a white matter disorder. So we'll see in a second if that is correct. All right, moving on to Rick's poll. Rick will present in a little bit about normal aging and Alzheimer's disease. And his question is, do you consider Alzheimer's disease a biological entity or a clinical syndrome? This is, I think, a hot topic still, Rick, but it's as long as I've, well, been in the field, this question has not been solved, really. So why is that, do you think?
Yeah, it's probably because there are sort of two perspectives still on this. So there's a stream of people that see it as a purely clinical syndrome. So you should be looking at the symptoms the patients experience. The other way of looking at it is more from a biological perspective. So we know that biologically the disease starts very early on, maybe 20, 30 years before actually symptoms start to emerge. So I guess if you want to treat the underlying etiology, it's also very important to understand the biology. So there are also ways, obviously, of combining it, but yeah, you should start with one. It's a biology or the clinic, and there's still a big debate about this.
That's right, but that's also why I think it's so nice to have you at the TN2 session, because it's about combining clinic and biology, basically. So, and most people think it's a biological entity. So, okay, I'm curious. So. Now we'll move on to the presentations, and afterwards we'll come back again to discuss the questions with you, and at the end we'll have some more polls. So I'll stop sharing my screen. I think we are good to go. Mike, are you ready?
Yeah, okay. So once again, many thanks for your kind introduction. In particular, many thanks for your kind invitations. Always very nice to go back to my roots in the Netherlands. So my presentation is about the MRI of opportunistic infections. So I already introduced the issue of immune modulation or immune suppression, treatment-related immune suppression as a potential cause of opportunistic infection affecting the central nervous system. So this is the agenda. So I start with some introductory slides. So what the definition is of opportunistic infection. Focus a little bit on how to diagnose opportunistic infection, not only based on imaging, but also using clinical presentation and CSF biomarkers. And then I will focus on PML as a sort of role model of CNS opportunistic infection, using the diagnostic criteria and introducing the heterogenic imaging finding spectrum, and also focusing on selective virus tropism, addressing the poll question we were just talking about, whether or not it's gray or it's white matter. And then I will end my presentation with a little bit of a look into the future, whether or not we can treat opportunistic infection, and what the role of imaging is in terms of treatment monitoring. So, what are opportunistic infections? So, opportunistic infections are basically infection caused by pathogens, can be either bacteria, viruses, fungi, or protozoa that take advantage of the impaired immune system. So, in a healthy host, these infections are not able to cause any clinical symptoms. So, the underlying immune suppression is basically the most important condition to identify opportunistic infection as real opportunistic infections. And the immune suppression conditions that are associated with opportunistic infections are the spectrum is quite heterogeneous and broad. So most of the data we have is dealing with the HIV infection. But in particular, cancer treatment, chemotherapy, organ transplantation, including bone marrow transplantation, but in particular, immunosuppressive treatments, so drug-related immunosuppression, plays increasingly a very important role because the spectrum of opportunistic infection might change based on the underlying immune suppressive conditions. So in other words, the spectrum of HIV-related opportunistic infection might be completely different compared to immune suppressive treatment-related opportunistic infections. So what is the spectrum of opportunistic infections? So in general, this is a large spectrum, a lot of viral pathogens. In particular, the GC virus can affect the CNS. We will focus on this more in a couple of minutes. Parasites, of course, toxoplasmosis, a very important opportunistic infection of the brain. Fungal pathogens like aspergillosis, cryptococcosis, and others, but also bacterial infections like tuberculosis and other pathogens that can cause an opportunistic infection even in the central nervous system. So it's very difficult, not only clinically, but also in terms of imaging, to identify the exact pathogen. And therefore, it's very important to realize that a diagnosis of opportunistic infection is based on three different aspects. So I always call it the holy trinity of diagnostic scheme. So it's the clinical symptoms, so the clinical presentation, imaging, of course, and the CSF assessment. And you should not exclusively interpret one of these aspects alone. So you should always consider the other two players. So in other words, you should not interpret the MRI without knowing the clinical symptoms and without knowing the findings derived from the CSF analysis. So therefore, the management and the diagnosis of CNS opportunistic infection is basically a three-step interactive algorithm. So all these three different modalities are interacting with each other. And this is also reflected by diagnostic criteria. So not only based on focusing on PML, but also on other opportunistic infections. And imaging becomes an increasingly important role because it's much more sensitive compared to the clinical presentation alone. I will focus on this in a couple of minutes. I told you that the spectrum of opportunistic infection is strongly depending on the underlying immune suppression. And my colleague and former PhD student, Martijn Wijburg, demonstrated this in a recent review article published in JNP, focusing on the spectrum of CNS complications, including opportunistic infection during immunosuppressive treatment of multiple sclerosis. And you can clearly appreciate that the spectrum is completely different compared to the HIV era. So in particular, GC virus-related diseases such as PML play a very important role, and they are quite frequent in these settings of immune suppression. So in other words, the underlying immune suppression gives us already an idea which CNS opportunistic infection we have to deal with. And in fact, PML has been getting increasingly important during the era of treatment related immune suppression. And this is reflected also by the number of published articles from 2005 on. You see an increasing number of case reports publishing PML as a role model of opportunistic infection based on drug-related immune suppression, and in particular, the immune suppression or immunomodulative treatment of multiple sclerosis plays a very important role in this setting. So PML is basically a role model of an opportunistic infection in the CNS, and as you know, we are dealing with a three-stage disease. So first of all, the latent, also the primary infection by inhaling the virus latency, which is the second stage in the kidney or in the bone marrow. And then when the patient gets immunocompromised, the patient can experience some reactivation of the GC virus, recombination of a rearrangement of the genetic material. And the GC virus can cross the blood-brain barrier and can cause an infection of the brain, in particular in affecting oligodendrocytes, leading to an irreversible demyelination of the white matter. But you see very nicely that also the gray matter can be affected. So it's not an exclusive white matter disease, although the terminology progressive multifocal leukoencephalopathy might suggest this. An MRI is basically able to identify these lesions very nicely with this bright signal on the T2 weighted images and on the dark signal reflecting the irreversible demyelination on the T1 weighted pulse sequences. So PML is defined as a sort of progressive multifocal leukoencephalopathy. Whether this still reflects the reality is another question. We'll focus this in a minute. So it's basically an encephalitis caused by the reactivation of GC virus, which is a papilloma virus. And it has a sort of selective tropism, mainly affecting the oligodendrocytes. So this lytic infection leads to an irreversible demyelination. And then it has another selective tropism because it does not affect the spinal cord and the optic nerve for reasons we do not understand yet. And it always, almost exclusively occurs in immunocompromised patients. So, and the problem is that still most of the data we have in terms of clinical findings, but also in terms of imaging findings, is derived from the HIV era. But as stated before, the treatment-related immunosuppression, in particular, patients treated with monoclonal antibodies like natalizumab or rituximab or other drugs, selectively addressing one specific pathway of the immune system is getting increasingly important. And I will show you in a couple of minutes that these conditions show a complete different phase of an opportunistic infection, in this case PML, compared to other conditions like HIV. So once again, PML, as other opportunistic infections, are diagnosed based on the Holy Trinity: clinical presentation, imaging, and the CSF. And this is also reflected by the diagnostic criteria. The four categories of diagnostic certainty: definite, probable, possible, or not PML, are based on these three modalities. In the case you have positive imaging findings, symptomatic patients, and you're able to demonstrate the virus in the CSF, you have a definite PML. In the case you have a patient without imaging findings, but still positive CSF and possible clinical findings, you have probable PML. But you might also face the situation that you are quite sure that you see a PML lesion on the brain and the patient is asymptomatic and you are not able to demonstrate virus DNA in the CSF. And according to these diagnostic criteria, we are not allowed to make the diagnosis of PML. Whether this is useful or not is another question, but this already reflects the problem that if we are quite sensitive in terms of imaging, without any supportive data from the CSF, we are not able to make the diagnosis according to this diagnostic criteria. So FLAIR, in terms of imaging, has the highest sensitivity in terms of detection of PML lesions. T2, conventional T2 is able to detect certain aspects of the pathology, like these punctual lesions or the vacuoles. T1 is able to show the degree of demyelination and also some signs of inflammation. And the DWI is able to show the active acute infection, and particularly in the border of the lesion where the lytic infection of the oligodendrocytes take place. So this is an example of the classic PML, a large lesion here in the frontal lobe, bright signal on the FLAIR in the T2, very dark signal on the T1. This is basically the pattern we know from the HIV era. And this is a typical lesion evolution. Once you see the lesion on the MRI, the lesion tends to growth. Once you are able to restore the immune system, you might see some inflammation representing the immune response against the virus, in particular, once again, in the border of the lesion. And in the case the patient survives the disease, you see some scarring and irreversible tissue damage. However, the lesion might also be quite small at the beginning, like in this natalizumab-associated PML, so a monoclonal antibody affecting the alpha-4 integrins or preventing the permeability of the blood-brain barrier, so no immune cells can cross the blood-brain barrier. And you see once again that the lesions can grow in the white matter following the white matter tracks, and you see very nicely these severe demyelination, irreversible demyelination in the white matter, reflected by this low signal on the T1. However, this typical white matter disease is not always the case, and not always the case is the absence of inflammation. So in this patient, you don't see any signs of inflammation. However, in monoclonal antibody, we can see inflammation quite frequently, reflected by contrast enhancement, abundance of enlarged perivascular spaces, reflecting an already present immune response against the virus, because the immune system in the CNS is not completely suppressed. And this is something completely different compared to the early PML findings based on the HIV area. And we call this finding the so-called sort of Milky Way appearance, which is quite specific for a monoclonal antibody associated PML. This is another example showing very nicely this contrast enhancement and looking at this contrast pattern. You would not think about PML. However, you see after the restoring of the immune system that the immune response is related to the inflammation we see initially on the inflammatory PML lesions. Another example, once again, these very subtle enhancement here in the extracortical white matter, not typical for PML if you're looking at HIV-related PML, but once again, if the patient gets a reconstitution of the immune system, you see that the immune response is really related already to the enhancement we saw initially on the MRI when the patient initially presented with the clinical symptoms. So inflammation is very frequent in monoclonal antibody PML, and we see another tropism of the virus. So we see a lot of gray matter involvement. So PML is not an inclusive white matter disease, and it can start already in the cortical gray matter, and it can spread in the cortical gray matter, and it gets disseminated, and secondarily involving the white matter. And it can also affect deep gray matter structures like here in the thalamus, and then secondly, getting connected to the cortical spinal tract and involving the cortical gray matter. So once again, moving to another spectrum of immune suppression, we are focusing on a different tropism of the virus, not exclusively affecting the white matter, but also the gray matter. And this is an example of a selective virus tropism, another GC virus entity called GC virus encephalopathy, which is characterized by an exclusive involvement of the cortical gray matter neurons. and the manifestations of the disease exclusively in the cortical gray matter. Another example of a selective virus tropism is the granule cell neuronopathy, exclusively infecting the granule cells of the cerebellum, very nicely shown here in this histopathology, and this leads to irreversible neuronal damage and atrophy in the cerebellum. So once again, moving to other underlying immune suppressive conditions, we are focused on different tropism of the virus, and it can even exclusively affect gray matter cells without affecting any white matter cells. So how sensitive are we with brain MRI? I showed you very large lesions, but if you screen patients with a higher risk of developing PML, you might see PML lesions with a very small lesion volume and being or remaining quite small in terms of the lesion dissemination. And this, once again, in terms of lesion morphology, has nothing to do anymore with the HIV-related PML with these large lesions in the frontal lobe and elsewhere. And that's the problem, because we know that imaging is so sensitive in terms of PML lesion detection that outperforms not only the clinical presentation, but also outperforms the sensitivity of detecting virus DNA in the CSF. And I showed you already the diagnostic criteria, and I introduced already the problem that that we can see PML lesions on the MRI without detecting any virus DNA in the CSF, and this can be present in approximately 20% of the patients. So once again, this can lead to a diagnostic problem, because MRI is so sensitive in terms of PML lesion detection, much more sensitive compared to the clinical presentation and the CSF analysis. And this sensitivity is based on the lesion volume. So we know that if you have larger lesion volumes, the risk of or the chance of detecting virus in the CSF is larger. So, but if you're moving to smaller lesion volumes, and I showed you if we were screening for PML detecting very small lesions, the chance that we can see any virus DNA in the CSF is becoming much lower. So therefore, once again, the MRI is the most sensitive diagnostic method in the detection of PML lesion or opportunistic infection in general. In terms of lesion evolution, PML associated with monoclonal antibodies can also show a very mild lesion evolution. You see that lesions starting here once again in the cortical gray matter need almost 6 months until they get disseminated in the contralateral hemisphere. So we see that PML can also be a mild progressive disease compared to the rapid progressive disease in HIV-related immunosuppression. And PML is also not always progressive. This is an example of a PML lesion here in the occipital lobe. Once again, treatment-related, when patients were switched from natalizumab, monoclonal antibody against alpha-4 integrin, to ocrelizumab, which is a CD20 antibody. And you see that these PML lesions are popping up, but you see that they stay the same, so they are not growing. They're sometimes shrinking after the immune response gets consolidated. So once again, PML is not always progressive, can also be quite stable in certain underlying immunosuppressive conditions. So finally, what do we have in terms of treatment? So if we have a monoclonal antibody associated PML, we can immediately restore the immune system by discontinuing the treatment. However, if we have a transplantation-related PML or a cancer-related PML, we have to offer the patient some treatments, helping to restore the immune system. And there are a couple of new strategies on the block right now. Checkpoint inhibitors like pembrolizumab, initiating the T-cell response and therefore able to clean the virus, or even the injection of allogenic BK virus or GC virus specific T cells that can attack the virus and help to restore the immune system. The next question is, do these treatments really help, and can imaging help to monitor these patients? And the answer is yes. This is an example of Pembrolizumab treatment, so a checkpoint inhibitor, and you can very nicely see on these images that after the injection of the checkpoint inhibitor, the lesion stops to grow. So the lesion is constantly in terms of the lesion, constant in terms of the lesion volume. So you don't see any further disease progression on the MRI, and the viral load on this in the CSF gets to zero. So the checkpoint inhibitor can stop this disease progression in some patients. And more nicely, this is the case in patients treated with allogenic BK specific T cells. Another BK virus is another polynoma virus, quite similar to the GC virus. And you see that after the injection of the T cells, you see that the lesion progression stops. You see even that the T2 lesions are getting smaller. And you see also that the inflammation related to PML is going to zero. So these are quite promising treatment strategies to help patients to restore their immune system and to stop PML disease progression. And I think imaging can play a role in monitoring these patients. Okay, to sum up, I try to show you that PML is a sort of role model of CNS opportunistic infections in terms of diagnostic workup. So once again, consider the Holy Trinity: clinical presentation, imaging, and CSF analysis. It's quite heterogeneous in terms of imaging finding, but also clinically. And it really nicely demonstrates the heterogeneity, but also the selective tropism of the pathogen, and that the selective tropism depends on the underlying disease or on the underlying immune suppression. PML or opportunistic in general can change their face, once again, depending on the immunosuppressive conditions. We see new imaging findings. We are very sensitive with our imaging findings, which requires apparently new diagnostic criteria. And we are able to show the lesion evolution, even showing PML lesions that are not progressive over time. And we have some new promising treatment strategy on the way, which might improve patient outcome. And the crucial question will be whether or not imaging can serve as an outcome measure for clinical trials treating these patients with these new treatment strategies. And this will be the next poll question. So once again, many thanks for your kind invitation, and thank you very much for your attention.
Thank you very much, Mike. This was a very interesting talk, and I'm also glad that you highlighted the answer to the poll. That is basically both, but it's still complicated. Before we move on to questions from the audience, I'd like to ask everyone, ask your questions in the Q&A button below. And in around 5 minutes, we'll get back to those questions. And for now, we invited Jenny Nijbijvank to join the discussion. So can you...
Now, do you want me to stop the screen sharing?
Maybe that's easiest. Yeah, I think so. Yeah. So Jenny is a postdoc at our MS Center and uses a lot of different types of imaging with a special interest in the visual system. So Jenny, go ahead.
Thank you, Menno. So thank you, Mike, for your very interesting presentation. So I was wondering, as you nicely showed in your presentation, it sometimes can be difficult to recognize PML on MRI, especially in early stages, and sometimes also to differentiate them from new MS lesions. And even though you told us that MRI is quite sensitive in comparison to clinical symptoms, if I can understand it correctly, it's still the cost-benefit ratio of like frequent MRI screening is not yet established. So I was kind of wondering, is there also maybe new research in maybe less costly screening markers, or maybe new imaging features or modalities that can even differentiate better between PML and new MS lesions?
Yeah, I think you're basically asking two questions. So first of all, whether or not we can differentiate lesions. So we are able to differentiate. So my colleague Martijn Wijbroek established some criteria helping to differentiate PML from all the other lesions, in particular MS lesions that might mimic PML. So in particular, the cortical gray matter involvement in the puncture pattern. So the inflammation I showed you is quite specific for PML, and we don't see this in MS. In MS, we frequently see periventricle lesions, and this is basically an imaging finding we don't see in PML, because PML is either in a deep white matter or in the juxtacortical white matter. The second question is a very tricky one, because so cost-benefit ratio is a very important issue. So I think the strategy changed a little bit, because in particular, if we're talking about natalizumab-associated PML, we're now facing the new era of the extended dosing. My colleague and friend Juppe Killestein performed a trial on this extended dosing, showing very nicely that the efficacy of the drug is still basically the same, but that the chance of developing opportunistic infection dropped dramatically. So I think that the frequency of opportunistic infection, having this extended dosing scheme is lower. So I think screening strategy should also be a different one in these patients. And to be honest, the screening strategy has only been established for natalizumab treatment. So it's not established for any other drugs. So the cost-benefit ratio does not justify any screening scheme for any other treatments. However, maybe some new treatment pops up in the future, once again being associated with PML, then we have to rethink the whole strategy again. But I think that the screening strategy, or the time of the screening strategy, is a little bit over, because once again we're going to an extended dosing, and the frequency of PML and other treatments is much lower.
Thank you.
Great, thank you, Jenny. Now we'll move on to questions from the audience, and to do so, Matthijs Brouwer will join us. He's a neurologist from our hospital and also an expert at neuroinfection. So Matthijs, go ahead.
Thank you, Menno. Excellent talk, Mike. There was one question from the audience that was also voted by on other members or panel, people listening to the talk, and that was whether once you established PML in a patient with natalizumab treatment, can you lower the dose, or is it necessary to switch to a different treatment?
Yeah, that's a good question. So I think, so once again, first of all, you have to restore the immune system completely, because you have to clean the CNS completely in terms of the presence of the virus. So you have to be sure that the immune system really clears the viral load in the CNS. And after establishing this, then you can consider a switch. My gut feeling says that's not a good idea to continue with natalizumab, even not with an extended dosing. So I would always prefer to, I would argue to switch to another treatment like SP1 regulator or a modulator or CD20 antibody.
Okay.
And I would not recommend to restart natalizumab again.
Okay.
However, there's no data for it, so it has not been done before. But it's only a gut feeling question.
Okay, and last year we've treated at our center 2 patients with PML due to a primary immunodeficiency with pembrolizumab.
Yeah.
Actually, they responded quite well even after 1 dose of pembrolizumab, which is different than was previously reported in the literature.
Yeah.
One problem we had was that we found it difficult to assess the MRI imaging whether the infection was still active or whether it was a remaining lesion. And you told me that the diffusion weight imaging may be able to show activity of the infection. Is that true? Can you?
Yeah. So it's tricky to identify PML in these patients because PML has 1,000 different phases, so it can show a lot of inflammation, and like I showed you in my last slide, it can also present as a classical PML. So first of all, it's very crucial to be 100% sure that this patient has real PML or not. So therefore, you always need to confirm your imaging findings by the CSF, I think, before starting the treatment. And then I think it's very important to identify the right patient for the right treatments. And I think if you would like to use pembrolizumab, you have to be sure that the T cells are not completely depleted. So you have to have a sort of functioning T cells in the patient. So not only a relatively high number, because but also you have to have a functioning T cells. Without a good T cell number and function T cells, pembrolizumab will not help you. And this is the same for allogenic BK specific T cells infusions. So therefore, the first step is to be 100% sure that this patient has PML, and I think the DWI is not always positive. So once you think about PML, you have to confirm the diagnosis by CSF or even by biopsy, because you don't want to treat patients with checkpoint inhibitors for these purposes not having PML. And the second very important issue is to look into the immune system of the patient, whether or not the patient has functioning T cells and enough T cells. This is also crucial to anticipate whether pembrolizumab will work or not.
Okay, well, thanks. I think we ran out of time, Menno.
That's right.
Well, yeah. So thank you very much for your answers, and well, Menno, continue with the next talk, I guess.
Thank you. Mike, thank you very much for your talk. Stick around because we'll come back to you as well after Rick's talk. So now I'd like to reintroduce Rick Ossenkoppele. As I mentioned, he is a neuroscientist working at our center, but he also has a position at Lund University in Sweden and has also been at UCSF. So he's traveled the world and has become an expert in the topic he will talk about, namely PET imaging of tau in Alzheimer's disease. So Rick, go ahead.
All right. Many thanks for the introduction, Menno. This is a bit late. I apologize for this. There we go. So thanks a lot, Menno, for the introduction, and also for the organizers of the TN2 meeting for giving me the opportunity to present today. We're living in a weird situation at the moment. I never expected to give a talk from the bedroom of my 4-year-old daughter, but yeah, it's the most quiet place in the apartment today. So the topics I would like to discuss today is to provide a little bit of a background on Alzheimer's disease, then highlight why I think it's very important to study tau. Then we are now able to actually image this in vivo using tau PET. And I will spend most of the talk focusing on research we have been doing using tau PET, and also to see whether there's a potential clinical application of tau PET. So first, a bit of background on Alzheimer's disease. So unlike most neurodegenerative disorders, Alzheimer's disease is characterized by 2 key neuropathological features, and they are amyloid pathology and tau pathology. Both of them are probably very important to the disease, but they act in different stages and also in different spatial patterns. So here you can see the progression of the disease going from cognitively normal to dementia. And you can see that amyloid pathology starts to develop really early on, almost plateaus already when persons are still cognitively normal, while you see that the tau pathology sort of progresses also when the clinical disease progresses. So that's important to keep in mind for the rest of the talk. So why would we want to study tau? Well, we know that abnormal tau correlates very strongly with disease severity and symptom progression measured ante-mortem. And we also know that it's linked to neural dysfunction and degeneration. However, until quite recently, all the knowledge regarding tau pathology and downstream processes were derived from animal and cell culture studies, from postmortem neuropathology, and from CSF measures. And being able to measure tau pathology using PET has many advantages. One of them, of course, compared to, for example, cell models or animal studies, is the ecological validity. So we're studying the disease in the species we eventually want to cure. We can study early disease stages. This is harder in, for example, neuropathological studies. With imaging, we can do longitudinal measurements, so we can really track the progression of the disease over time. And a very big advantage, I think, of PET over CSF is that you can do very detailed mapping of the entire brain. So in CSF or in plasma nowadays, you get a single number representing the entire brain. With PET imaging, you can see where in the brain the pathology occurs. So moving on to tau PET. So it's a relatively recent technique. So we have been able to measure amyloid pathology with PET for decades now. The first tau PET publications came out around 2013. On the left, you see an autoradiography study with one of those tracers called fluortauxipir. And you can see in a person that was positive for amyloid beta, but negative for tau pathology, you can see that there is no binding of the tracer to tau pathology. Whereas if we do the same experiment in a person that is amyloid positive, but also tau positive, you can see binding of the tracer in the gray matter areas. So it's specific to tau and not to amyloid beta. You always have to check whether it is also the case when you inject a PET tracer during life. And this was established in this study where you see the amount of tau PET uptake on the x-axis and the amount of postmortem pathology on the y-axis. And you can see that in those same regions, there's a very strong correlation between antemortem and postmortem. And this relationship was completely absent for amyloid beta relative to tau pathology. So again, it's only tau PET that relates to postmortem neurofibrillary tau tangle tau pathology. So moving forward now to 2021, we have now more or less 4 tau PET tracers that are now widely used, most of them in research, but one of them also in clinic. I'll talk about that later. And when we look at group averages from cognitively impaired individuals, you can see that across these tracers, there's a very common pattern that emerges. So there's a very high retention in the lateral temporal parietal cortex. There's also uptake in the posterior cingulate and the precuneus area. And also the medial temporal lobe gets affected together with the inferior temporal cortex. It's also very easy to distinguish the images from the cognitively impaired individuals. So they had MCI or Alzheimer's disease dementia. And you compare them to cognitively unimpaired individuals. You can see that there's barely any tracer uptake. So the color scale indicates the amount of retention going from low to high. So in terms of research, I had to be very selective, but I chose 2 topics. The first one is atypical variants of Alzheimer's disease, because like in the previous talks, heterogeneity plays a really big role also in Alzheimer's disease. And I think it can inform us a lot about how the disease works and which pathologies play an important role in, for example, the manifestation of symptoms. And I selected the second topic, so modifiers of tau progression on PET, because I think it has a lot of therapeutic potential. So I think if we think tau is an important target, and I think that's the case, then halting or stopping the progression of tau can hold a lot of therapeutic potential. So let's start with the atypical variants of Alzheimer's disease. And I'm starting here with a case that was diagnosed with posterior cortical atrophy. So this is a very rare phenotype, and it's characterized by visual impairment. So in general, Alzheimer's disease is mainly characterized by memory impairment. In this specific phenotype, people have symptoms like trouble in, for example, orientation, identifying faces, finding the way, having trouble getting dressed, for example, so all sorts of visual spatial deficits. So this is a case study of a person that underwent 4 imaging modalities. So the first two here are shown already. First one was with FDG PET, where we measure hypometabolism. And if you look at the red arrows, those are areas that are hypometabolic. So those are regions that show insufficient uptake, indicating that the level of glucose metabolism is relatively low there. We also did an amyloid PET scan. So it's clearly a positive scan. So the red areas indicate where there's a lot of amyloid pathology in the brain. But you can see it's all over the brain. It's both in posterior brain areas, but it's also in frontal areas. And when we look at the tau PET scan we did at that time, you can see that it's almost a mirror image of the FDG PET scan. So in regions where there's a lot of tau, you see a hypometabolic pattern. You see it all the way here. And in regions where there's not so much tau, the glucose metabolism is relatively preserved. So there seems to be a very strong correlation here between the presence and the spatial pattern of tau pathology and where neurodegeneration occurs. So this was just a case study. So we did the same thing when we included 10 patients with posterior cortical atrophy, and we essentially found the same thing. So when we looked at amyloid PET using an amyloid PET tracer, we found that there was essentially PET tracer uptake throughout the brain, including frontal areas, whereas with the tau PET tracer, you can see it's really restricted to those posterior areas of the brain. And we know those regions are very important for those visual spatial functions. So the same also applies to other phenotypes of Alzheimer's disease. So for example, in logopenic variant primary progressive aphasia, also known as language AD, you can see that there's much more tau in the left hemisphere compared to the right hemisphere. And we know that a lot of our language function are orchestrated from the language dominant left hemisphere. In an amnesic syndrome with memory AD, you can see that tau is really concentrated in the medial-temporal areas. And in cortical basal syndrome, which can lead to, among others, motor symptoms, you can see that there's very uniquely tau PET tracer uptake in the sensory motor areas. And we don't see this in any of the other variants of Alzheimer's disease. So the conclusion based on these phenotypes is that tau PET patterns mirror clinical heterogeneity in Alzheimer's disease. There's one exception, that's the behavioral variant of Alzheimer's disease. So those people show really predominant behavioral deficits like disinhibition, making social inappropriate jokes, for example, being compulsive, showing dysexecutive symptomatology. But when we do tau PET in those cases, we do actually see quite a posterior pattern, which you may not expect based on the clinical symptoms. So we are still trying to understand this disease a little bit better. It's work by our PhD student, Ellen Singleton. And if you want to read more about this, you can look into this publication. Second part is modifiers of tau progression with PET. And the first step here is to actually establish whether tau PET signal does change over time. So you can see a selection of studies here, longitudinal tau PET studies, and you can see that all of them are relatively recent. So the longitudinal data is just starting to get in. I think a general observation from all these studies is that yes, there is an increase in tau pathology over time as measured with PET. It's relatively low in people that are cognitively normal. So it's somewhere between 0.5% and 2 to 3%. And in symptomatic stages of Alzheimer's disease, it is somewhere between 3% and 10% for every year. So also the more advanced the disease stage, the higher the rate of accumulation of tau pathology. So now we have established that tau does actually change over time, which we obviously expected based on a lot of literature. It's interesting to look at potential factors that can modify the rate of tau progression over time. And the first one I'll discuss is amyloid beta pathology. So one potential source of evidence is that amyloid beta is related to spreading of tau, is that if we look at the data and we sort of binarize the amyloid status and the tau status, you can see that it's extremely rare to be amyloid negative, but be tau positive. This was only the case in 5 out of 700 plus cases in the study, whereas the opposite is much more common. So there are a lot of cases that are positive on both. The opposite is also often true. So there are many cases that are amyloid beta positive, but are tau negative. And the expectation is that they may become tau PET positive if you follow them for a longer time. So tau PET positivity almost exclusively occurs in the presence of amyloid pathology. Second, this is a pretty complicated schematic figure of a mediation analysis, but this was a longitudinal study with multiple time points, including amyloid PET and tau PET and also cognition. And the take home from this study is if you do progress in terms of amyloid beta pathology, there's also a change or an increase in tau PET pathology. In other words, also if you're stable in terms of amyloid beta pathology, so you do not accumulate amyloid beta pathology over time, then also the levels of tau PET are stable. And finally, we know from the neuropathological literature that there's a very specific condition called PART. It's primary age-related tauopathy. So this is a tauopathy that occurs without the presence of amyloid beta pathology. But if we carefully inspect the spatial distribution of this tau PET pattern, we can see that it's relatively limited. So it goes into the temporal cortex, the basal forebrain, and the cingulum, but it doesn't spread to the rest of the cortex. Whereas in Alzheimer's disease, where both of those proteins are present, you can see that only in those cases, when amyloid beta is present, you can see that also other parts of the neocortex get involved. Another potential modifier is age. And what we previously already have shown is that younger persons are more affected by tau pathology than older persons. So if we do a direct comparison between people with early onset AD, they're younger than 65, and late onset Alzheimer's disease, they're older than 65, you can see that the young group has more tau pathology, especially in neocortical areas like the parietal cortex and also the lateral frontal areas. And what we recently showed is that also in terms of change over time, so you can see the tau PET slopes here on the y-axis, that the younger people are, the more tau they accumulate over time. And this gets lower with increasing age. There are also some very interesting sex differences. So for example, what was previously already shown that at similar levels of amyloid pathology, females tend to have a higher tau PET signal. And we also previously showed is that females had higher brain resilience. So despite the fact that they have more tau pathology in the brain, they're better capable of preserving brain structure, like, for example, the cortical thickness. And we also recently showed in the same study that females accumulated more tau pathology over time compared to males, even when we corrected for amyloid beta status. And finally, I think a very interesting one, which really nicely connects clinical with basic neuroscience, is the role of potential connectivity. So it has been shown that tau spreads in very stereotypical patterns throughout the brain. And basic science studies have suggested that this may occur across connected regions. So there have been many attempts to make the link between basic science and clinical neuroscience. This is a study we performed. It's a cross-sectional study, and we looked at a group of 63 patients with Alzheimer's disease, and we looked at the tau PET covariance. So when we placed a seed in the occipital cortex, we found that across all those patients, those were the regions that covaried with the seed. So in other words, those are the regions that showed high tau PET uptake compared to the levels in that particular region. And if we compare that exact same analysis, but now using functional connectivity in healthy young adults as the outcome, you can see that these patterns are really remarkably similar. So in other words, the way tau PET spreads throughout the brain is very closely linked to the functional architecture of the brain. But of course, this is just a cross-sectional study. So we also did the same thing, but then using a longitudinal design. So on the y-axis, you can see the change in tau PET. This is the connectivity strength. You can see that the higher the level of connectivity, also the higher the rate of accumulation. So this is across the whole brain, where we found the same thing in specific brain networks, like the default mode network, the limbic network, and also a visual network. So to summarize, the functional brain architecture is associated with the rate of tau accumulation. And then finally, is there potential for tau PET in the clinic? So we set out to study this. We studied 4 different groups. So we had cognitively normal people, we had people with MCI due to AD. So these people have objective impairments, but it's not severe enough to call it Alzheimer's disease, but they do have abnormal amyloid biomarkers. We studied a group of people who had Alzheimer's disease dementia and were also amyloid positive. And then a big group of people that had non-AD conditions, for example, frontotemporal dementia, vascular dementia, dementia with Lewy bodies, etc. And just by looking at the images, you can already see that there's a really big difference between people with Alzheimer's disease dementia and people with another type of dementia. When we actually quantify this difference, so we extracted the mean uptake in a specific region of interest. So you can see the value here, the higher, the more tau. And we established a cutoff, that's a dotted line here. So if we look at the group with AD dementia, you can see that really the vast majority of them are considered tau PET positive. So they have a value that is above this cutoff. Same is true for the group with MCI due to AD, only to a lesser extent. So about 2/3 of this group is considered tau PET positive. But then if you look at all the other groups, really the vast majority of them are tau negative. So the tau PET positive cases are really an exception. So if you then translate this into diagnostic numbers, you see that the area under the curve is 0.95 and a specificity of a sensitivity and a specificity of around 90%. And this is a major improvement compared to the clinical diagnosis, which is typically around 70% for both. We also tried to test whether the performance of tau PET would be better or equal to established markers of MRI. So we performed area under the curve analysis. These fixed lines are the slopes for tau PET in different regions of interest. Those are the lines for MRI measurements. And you can see that the difference in area under the curve is quite big. And this is the area under the curve for discriminating AD dementia versus other types of neurodegenerative disorders. So the gap is pretty big and really high, as stated before. But there's also a big drop when we move to earlier stages of the disease. So you can see that the area under the curve is really much lower compared to the AD dementia stage, although it's still quite much better than MRI. So these and other studies have also now resulted in actual FDA approval of fludarabine. So the first tau PET tracer we used for the clinical use in people with Alzheimer's disease dementia or are suspected of Alzheimer's disease dementia. So this is really a big step forward for the field and has been achieved in a relatively short amount of time. Then is there also potential for prognostic use. So this is first a cross-sectional study where we looked at 3 different markers, so tau PET, amyloid PET, and MRI, cortical thickness. We looked at a different region of interest. You can see them also here. And we looked at a variety of cognitive tests, including global cognition, memory, processing speed, and language. And what we can see if we look at the preclinical stage, so those are persons that are cognitively normal but are amyloid positive, you can see that there are only significant associations between tau PET and cognition and not with the other modalities. Looking at the symptomatic stage, you can see that there are significant associations both for tau PET and for MR, probably a bit stronger for tau PET than for MR, but amyloid PET is not related at all to the cognitive performance. So this is ongoing work. I'm pretty excited about this. So we collected a pretty big data set of about 1,400 individuals across the whole group. We calculated change in MMSE, which is a measure of global cognition. So this is the annual change on that measurement. And we looked at the association between tau pathology measured on PET in a specific region of interest and compared that against an MRI measure. And we look at the whole group. You can see that the proportion of explained variance is higher for tau PET compared to MRI. And this is especially pronounced in the early stages of Alzheimer's disease. So when we look at the group of amyloid positive mild cognitive impairment, you can see that the R-square is much higher for PET than for MRI. And the same is true for individuals with preclinical Alzheimer's disease. So in summary, tau PET retention corresponds to many of the neuropathological findings. Tau, but not amyloid beta PET patterns mirror sites of neurodegeneration. Tau pathology may be the driver of cognitive loss and of distinct variants of Alzheimer's disease. We can detect meaningful changes in tau pathology using PET. Tau PET has now been approved by the FDA for diagnostic use at the dementia stage. And I think tau PET has great potential for prognostic use in more early stages of Alzheimer's disease. So I would like to thank many of the collaborators that I worked with over the past couple of years. And I promise my colleagues from Hersenonderzoek to have a quick shout out. So if you are looking for participants, there's a really big database available now, including more than 22,000 people that are really excited to participate in research. So if you're looking for people, you can either check out a website or send an email to this address. And that was it. Thanks for your attention.
Thank you, Rick. Very great presentation and a lot of new developments. It's really, really exciting. What we did now is we invited Laura Jongman to join the discussion. Laura is an assistant professor at the Amsterdam UMC and has a very broad interest in combining pathology with MRI and also with a network interest. So Laura, go ahead.
Yes, thank you for the introduction. Well, mostly thank you, Rick, for this very interesting talk with, showing the added benefits of PET imaging in both aging and AD. And so you also showed some tau PET related in neuropathology, which associate together slightly less with amyloid PET. And I know from the literature that some studies show that PET only picks up at later Brach neurofibrillary tangle stages or later tau amyloid phases. So I was wondering if you think that PET will eventually improve or develop itself to also pick up the very earliest stages of these pathological accumulation, or what are the challenges are in doing so?
Yeah, thank you, Laura. Very important question, of course. My question at this point with the currently available PET tracers is that they are not that sensitive, unfortunately. So there are a couple of studies now that looked at antemortem and postmortem relationships. And what we can see is tau PET is doing a really good job when people are in Brach stage 5 or 6, or really end stage pathology, but way worse in especially Brach stage 1 to 3. So it's really not picking up the pathology there. So I think there are pros and cons to that. I think a big negative thing is, you would want to be able to detect the disease as early as possible. Potentially the good thing about it is that Brach 1 to 3 is probably not enough to cause a dementia syndrome. So if you look purely at like diagnostic purposes, I think it's actually a big advantage that we're actually way more specific, and that has always been a big problem with amyloid PET. So if you do an amyloid PET scan and it's positive, you never know whether Alzheimer's disease is really the primary pathology. With tau PET, if it's positive, it's really highly likely that's the case.
Yes, and then are there any developments to make PET more sensitive?
Yeah, so most of the studies I've shown today are with the first generation tau PET tracer called Flotausapeer. You may remember that one of the first slides I showed with those 4 different tracers. So the other 3 of them are so-called second generation tracers, and they are potentially a bit more sensitive. They also have a bit less problems in terms of the medial temporal lobe region. So Flotausapeer has a lot of off-target binding in the choroid plexus that is really close to the hippocampus. So the signal we see in the hippocampus is not completely reliable with Flotausapeer. It's potentially more reliable with these other tracers. But yeah, those tracers have been introduced just a couple of years. So those bigger diagnostic and prognostic studies have to be performed yet.
Great, thank you.
All right, thank you, Laura. Then we'll move on to questions from the audience, and Jeroen Hozemans, expert in neuropathology of Alzheimer's disease, will join the discussion for that. Go ahead, Jeroen.
Yeah, Rick, thank you for this very nice presentation. We also have some questions from the audience, starting off about the the tau tracers. You already talked about the second generation of tau tracers, and Obada has a question whether there's any hope for an antibody-based tau PET tracer, or are there already developments in that?
Yeah, during the coronavirus period, I compiled an impressive list of papers that I have to read. And one of them is actually like a preclinical study on antibody-based tau PET tracer. So I still have to read that paper, so I can't sufficiently answer the question. But the thing I know is that people are working on it.
Okay, thank you. And another question from Abada: are the PET tracers, can they also be used for routine diagnostics in the clinics, or how's your view on that?
Yeah, so actually, I've just been working on an opinion paper on that. Answer is yes, with the nuance that it needs to be done very selectively, and also only in people with dementia. So I think I showed before and also discussed with Laura that there is a lack of sensitivity. So I think it only adds a lot to existing biomarkers at the dementia stage. So in the United States, it's actually already been used because it has been approved by the authorities. It's not yet the case in Europe, but I think that the results showing that tau PET so far, again at the dementia stage, differentiating AD from frontotemporal dementia, vascular dementia, dementia with Lewy bodies, etc., are that impressive that I think that they will be selectively used in the future.
And then there's a question from Gert about the correlation between the NERP psychological characteristics in AD variants and the tau PET levels.
Yeah,
is there a correlation?
Yes, there is. So across the board, so regardless of clinical phenotype, the amount of tau matters, but also the location matters. So for example, if you have more tau pathology in the left hemisphere, you're also more likely to have language deficit, for example. If the tau pathology is really more posterior, you will find more visual-related impairments. And if it's in the medial temporal lobe, then you're most likely to have a memory predominant phenotype. So those relationships are very strong, except with what I showed for behavior. So the phenotype with behavior, we really don't fully understand yet, but we're working on it.
Yeah. And maybe we have time for one last question, also a question that I had and also Arjen posted in the Q&A. So we know from in vitro studies and vivo studies that A-beta pathology depends on expression of tau. How do you see that in living person? Are there also studies done on a combination of tau PET and amyloid PET? Are these combined, and do these studies also give some idea on how actually A-beta and tau interact during life?
Yeah. Really important question, of course. The issue, and I think I started my presentation with this. There are two issues with amyloid and tau. One is that amyloid, at least in the neocortex, starts to accumulate much earlier than tau pathology, potentially decades earlier. And second of all, they tend to accumulate in different areas. So that raises a lot of questions. If one is related to the other, why do they not occur in the same place? And why is there such a big temporal gap between them? And to be very honest, I don't think imaging studies can provide insight into mechanisms there. We can only do sort of association studies. So we've been linking some sort of like gene expression profiles, for example. And one sort of overlapping process there is lipid metabolism. So in areas that are expressed in both show up there. Whilst if you look at tau PET specific patterns, it's more exon related, and if you look at amyloid PET patterns, it's more dendrite related. So that's one potential mechanism. But again, I think this is where we should hand our data over to more basic neuroscience and look further for mechanisms.
Okay, thank you. Looking back at Menno, I think I give the word back to you.
Okay, thank you,
Yes, thanks.
Jeroen. Thanks. Well, we're almost at the end of the show, as we say, and we have time for 2 more polls. So I'd like to start first with the poll of Rick, because it's still fresh in our memory. If we can have it on screen. This is a question that Rick himself thought of. Is Tau PET ready for clinical application? So did Rick convince you on the value of Tau PET? And Rick, does this mean, because you asked this question specifically, that amyloid PET is out the window for clinical application in that sense?
No, and especially not for the earlier stages. But as also came up during the discussion, the problem with amyloid PET, especially at older age, is I think it's most informative when it's negative, because then you can rule out that it's Alzheimer's disease. If it's positive, there's still always doubt whether it's really Alzheimer's disease that's ongoing, or whether the amyloid is sort of comorbid to something else. We see a lot of cases that have, for example, frontotemporal dementia or vascular dementia that in addition have amyloid pathology.
And also the paradox that you said that it doesn't relate well to symptoms is also, I guess, then tricky.
Yeah, but I'm not sure if that's really a problem for diagnostic use, right? So,
no.
But for
science?
For science, yeah. for sure, absolutely, yeah.
No, but close is the answer. Okay, but it's a close race. Some people also said yes, fully ready. So, what do you think yourself?
Yeah, I'm probably more to B.
Okay.
I think we're really close, but there are still a lot of open questions. We also still haven't fully characterized whether the tracer is binding to. You know, we haven't studied more representative populations with older people, for example. So there are some really important questions, I think, that should be answered before we can really apply it more routinely.
Yeah, so you still have some work to do. That's also...
Definitely, yeah.
All right, thank you. Now, coming back to Mike, we have a closing poll for him as well. Will imaging be a primary outcome measure in clinical treatment trials focusing on opportunistic infections? So did Mike convince you that imaging is a primary outcome measure and not only clinical features? Is that correct, Mike? Am I saying
Yeah,
right?
and the reason that I ask you guys this question is because I'm part of the FDA NIH group on clinical trials and PML, and we had a huge discussion whether or not we should include imaging as an outcome measure in clinical trials. So the more conservative ones are saying no, no way. And the more progressive ones are saying, yeah, we should consider it. And that was the reason that I tried to get your opinion on this.
I think also it's a problem across neurology, right? That in all treatment trials, imaging is still not accepted.
Yeah, I'm very pleased to see that the majority of you guys are on my yeah, it is an ongoing discussion, and we'll see what the future brings. But I think I showed you that imaging is quite helpful in monitoring the disease burden, in particular if you're treating with checkpoint inhibitors or with allogenic T cells.
Yeah, great. And Rick, in your field, is the same question also a problem? Are the PET tracers not primary endpoints yet?
So at the moment, they are mostly used for participant selection. So there's been a big problem in phase 3 clinical trials where about 20 to 30% of the participants who were treated with anti-amyloid drugs were amyloid negative. So it's really mandatory now to make sure that they actually have the pathology that's being treated. In most clinical trials nowadays, it's a secondary outcome measure. And it's often combined with MRI and sometimes also fluid biomarkers. But most trials now include it as well, but never as a primary.
No. And Mike, if you convince the committees that they should include imaging, but what would they choose? What is the best imaging marker as a primary?
Yeah, that's a very good question. So because that's tricky, because if you monitor the disease, you want to monitor, first of all, the disease progression in terms of volume. That would be the task for FLAIR T2. but you would like also to monitor the immune response, and the immune response is reflected by contrast enhancement, and therefore you need, I think, a combination of FLAIR-T2 demonstrating disease progression in terms of lesion load, and T1 post-gadolinium monitoring the immune response, which is reflected by inflammation on the MRI.
Yeah. Well, I hope you convinced them, because I think it would open a lot of doors.
Yeah. I'm working on it.
Yeah, great. So thank you to both of our speakers. And just to close this session, a final reminder that we have a few more talks coming up on the 18th of February, Jan Badma and Preyas Tewari, and then 2 more sessions after that. And if you want to register already, please go to our website to do so. And follow us on Twitter and LinkedIn. And thanks again to both of our speakers, and hope to see you next time.